So the topic for this video, the revision topic for this video is mode of action of antibiotics through the inhibition of specific enzyme systems. First kind of antibiotic here are the sulfonamides. Sulfonamides, the basic structure of sulfonamide include the p-aminobenzene sulfonamide structure. It contains a benzene ring with amide group, amino group attached to it, along with the sulfonamide group attached at the para position to each other. For the mode of action, it's known that bacteria require p-aminobenzoic acid, which is also known as PABA, as a precursor for the synthesis of some essential enzyme that is called THFA or tetrahydrofolic acid. The PABA is a structural part of THFA, and here sulfonamide molecule is similar in structure to the PABA molecule. So what happens is that the sulfonamide enters the reaction in place of PABA and it blocks the synthesis of THFA in the cell. So due to which it inhibits the growth of cells that synthesize THFA from PABA. And the sulfonamide, it does not interfere with the growth of cells requiring any folic acid, and it directly reduces it to THFA as in microbial cells, as well as it includes the mammalian host cells also. The action of sulfonamide is an example of competitive inhibition because um, the sulfonamide is structurally similar to PABA molecule, so there is a competitive inhibition between the essential metabolite PABA and the metabolic analog, that is sulfonamide. So here we can see in this diagram, it is depicted that how it will be acting. So in first case, pteridine will react with PABA, and in the presence of enzyme dihydropterate synthase, the DHPA will be formed, which upon reaction with L-glutamate, DHFA is formed, and which in the presence of dihydrofolate reductase will form the THFA enzyme. That is important for any microbial system. In case of B, where sulfonamide is added, pteridine will react with sulfonamide instead of PABA. Since sulfonamide and PABA are structurally similar, so sulfonamide will connect with uh, pteridine, and it will form a pteridine sulfonamide complex, which will not react with the L-glutamate, and their union will be blocked, and hence the THFA enzyme will not be formed, and hence the action of the system will be blocked. This is how the sulfonamide will act against the microbe. Moving on to antifungal antibiotics, the first one is nystatin. It is obtained from Streptomyces neusei and it's used for the therapy of non-systematic fungal infection. It was first discovered by Rachel Brown and Elizabeth Hazen in 1950. It's a polyene kind of antibiotic, and it's fungicidal in nature. The spectrum of antibiotic activity of nystatin include yeast and other fungi such as Candida, Aspergillus, Penicillium, and Botrytis. So the action of nystatin is restricted to yeast and fungus. The next one in this category is griseofulvin, which is obtained from Penicillium griseofulvin, and it is used for the treatment of superficial fungus infections of the body and the skin surfaces basically. So topical application is there, and in some cases it is also used for the treatment of some systemic mycosis which are deep seated. It can be administered orally. Moving on to antiviral chemotherapeutic agent, the first kind is the interferon. The interferons are chemotherapeutic agents that are being used for treating the viral diseases. They are small glycoprotein substances. When the cells that are exposed to interferon, they develop antiviral properties, and the action is involved in the interference through these interferon by interference of protein synthesis. There are two kinds of interferon mostly, leukocytic and fibroblasts. The next one in this category of antiviral chemotherapeutic agents is acycloguinosil. and it's a nucleoside analog. The acycloguanosine is a nucleoside analog, and it is active against the herpes virus in animals. It acts through the inhibition of nucleotide utilization. It utilizes the nucleotide and it inhibits the herpes virus in animals. The next one in this category is 5-iododeoxyuridine, and it is a synthetic analog of the pyrimidine nucleotide basically, and it acts by the inhibition of nucleotide acid synthesis. So it will not allow the nucleotide acid synthesis in the organism, in the virus, and it will prevent the incorporation of thymidine residue into the DNA. Next one in this category of antiviral are the amantadines. They are low molecular weight compounds effective against influenza A virus, and they are not effective against the influenza B virus. They act through interfering with the uncoating of the viral particles and the subsequent release of nucleic acid. So when they inhibit the uncoating of viral particles, so the nucleic acid is not released into the host organism, due to which the virus cannot affect the host organism. This is how amantadine will work. Next is the anti-tumor antibiotics. The first one is the anthromycin group, which includes anthromycin, cipromycin, tomamycin, neothromycin, both A and B types. The complicating factor that is a kind of a limitation for the use of anthromycin group includes the cardiotoxicity that they have, so they have limited use. The anti-tumor action of these anthromycin group is directed towards the DNA structure and function. So this was it for this video. I hope it helps. Thank you so much.